The confocal microscope was invented in 1955 by Marvin Minsky. The confocal microscope uses laser light to scan samples that have been dyed. This is very different from a light microscope for image formation. The samples are dyed and are prepared in slides and inserted. Then, with the aid of a dichromatic mirror, the microscope produces a magnified image on a computer screen. Anyone can create 3D image as well as by assembling multiple scans. The confocal microscope offers high degree of magnification and better resolution. This is used in cell biology and medical application, especially in studying live human cells. Today, there are 3 types of confocal microscope: a laser scanning microscope, a spinning disc confocal microscope, and a programmable array microscope, or PAM for short.
The main components of the confocal microscope are a detector, confocal pinhole, dichroic mirror, illumination pinhole, laser, objective lens, and focus. The main component that differentiates a confocal microscope from others is the confocal pinholes. The microscope works by taking a laser as a light source, and it will struck a wavelength from the dichroic mirror. The dichroic mirror is also referred to as a dichroic beam splitter. What this is a piece of glass with a thin layer of metal covering it. This will cause the wavelength to bounce off through an objective lens and interact with the specimen. Simultaneously, another wavelength is going to bounce off the specimen, and that wavelength will pass through the dichroic mirror and go through the confocal pinhole. Then the wavelength will proceed to hit the detector, thus allowing us to obtain the images. An advantage to this is that if any other light source were to come through, it might pass through the dichroic mirror, but it will not be able to pass through the pinhole. The aperture of the pin can be adjusted to either let more light through or decrease the amount of light that goes through. The confocal microscope uses software to operate. The software acts as an extension to the microscope to provide features that allow for the advanced image processing, analysis, and visualization of the images captured.
The confocal microscope is an ideal tool used in the field of developmental biology due to its outstanding optical quality and cell-friendly imaging, which provides an ideal platform for microscopic imaging. Genetic researchers also depend on this essential tool when observing protein expression and protein translocation. In the field of neurobiology, a combination of confocal microscopy, optical sectioning, and 3D imaging allows scientists to view cells of the nervous system deep into brain slices and tissues. Confocal and fluorescence microscopy is an essential tool for researchers studying cell signaling because it allows them to analyze tissues and cells to identify molecules of interest.
Software allows the recording of real-life imaging over time and the generation of 3D images or videos. Specific settings when using the microscope can be programmed through the software so that with the click of one button, all the necessary parameters will be set automatically.
A use for a confocal microscope in a clinical field can be the Reflectance confocal microscope. A Reflectance confocal microscope is a novel, non-invasive technique for examination of the skin. It provides real-time confocal images of a tissue as shown in the clip. Light will pass between the cellular structures, and it can capture and recompose into a 2-dimensional grayscale image on a computer software. The Reflectance confocal microscope can show images with enough detail for the use of histological analysis. There are 3 levels that can be observed: stratum corneum, superficial layer, and basal layer. Now that we know a little background of the Reflectance confocal microscope and how it works, let's look at how it could actually be used. The Reflectance confocal microscope is a tool of interest today on being used as a diagnostics tool. A case study explains on how Reflectance confocal microscope can be a potential use for skin cancer imaging for clinical use. Expert dermatologists can diagnose correctly around 75 to 80% of skin tumors. Final diagnosis can lead to surgical biopsy, and this can be painful, leave scars, and is time-consuming to have skin prepped for examination for diagnosis. The use of the non-invasive diagnostic techniques, such as the Reflectance confocal microscope, have been developed. The Reflectance confocal microscope can provide additional information that is more advanced in clinical inspection, can preserve the tissue, and provide real-time diagnostics. The Reflectance confocal microscope has the proper resolution that is necessary for seeing details for histological analysis and could be an alternative for physical dissection of the skin. A case study was done to see if the Reflectance confocal microscope can be a tool of interest to look at possible patients that could have actinic keratosis. Actinic keratosis is a precancerous lesion of the skin that is described to look rough, patchy, and scaly, caused by years of sun exposure. In this study, there were 7 patients with clinically diagnosed actinic keratosis and were enrolled in the study to use the Reflectance Confocal Microscope. The 7 patients included 5 men and 2 women from ages 47 to 75 years of age. The lesions of examination were on the hand of 3 patients, and the dorsal forearm of 2 patients, and on the forehead of 1 patient. The Reflectance Confocal Microscope took imaging of these lesions. The results show that 7 lesions of the skin were clinically diagnosed as actinic keratosis. In the figure shown, displays the results obtained in this case study. In the figure on the right column shows normal skin images using the Reflectance Confocal Microscope. The center column are diagnostics of hyperkeratosis, which is the thickening of the skin's outer layer. Lastly, in the left column shows a detailed morphological characteristics of the skin that depicts abnormality. From this study, it can be determined that the Reflectance Confocal Microscope can be an alternative biopsy and diagnosis of actinic keratosis. The case study gave an idea on how the Reflectance Confocal Microscope is used in a clinical setting. The Reflectance Confocal Microscope has many advantages. The Reflectance Confocal Microscope is a reliable alternative for a non-invasive technique and allows for investigation of a normal to a pathophysiological processes with high microscopic resolution that can be viewed in detail. If we combine the Reflectance Confocal Microscope with other non-invasive technologies, for example ultrasound or MRI, it can improve resolution and details. There are still some adjustments that need to be made for the future use, but once it is done, it can be a better alternative for imaging skin for clinical use.